So I discussed three trials, one in HCC and two in cholangiocarcinoma. The trial in HCC is a trial that tested in the first line in Asian population the combination of Tislelizumab, that is an anti-PD-1, plus Bevacizumab biosimilar, plus a new anti-TIGIT, so a new antibody against a new checkpoint inhibitor, versus the combination of Tislelizumab plus Bevacizumab biosimilar. Unfortunately, the trial is negative, but the double combination showed an interesting response rate of 37.5% with a short duration, 10.6 months, so we will need to see if we will be able to see prolonged duration of response maybe in the future. So the trial is negative, but there is still a rationale in using anti-TIGIT antibodies in HCC, and there is an ongoing phase III trial with another molecule that will be clarify if TIGIT inhibition has a role in HCC. Then the other two trials were in cholangiocarcinoma. The first one is a post-hoc analysis of three randomized trials, ABC-01, 02, and 03, that tested the role of gemcitabine cisplatin in first line in the advanced setting. This post-hoc analysis aimed to evaluate the outcome of patients with extrahepatic cholangiocarcinoma. This is important because we have now identified some molecular alterations with potentially targeted agents for patients with extrahepatic cholangiocarcinoma, like HER2 amplification. So it's important to have an idea of the outcome of these patients. And what this analysis clarified is that the prognosis of patients with extrahepatic cholangiocarcinoma is worse compared to the prognosis of patients with intrahepatic cholangiocarcinoma, but quite similar to the overall patient population with biliary tract cancer. So this data will be useful in the future to better understand the results of targeted agents against molecular alteration in patients with extrahepatic cholangiocarcinoma. The third trial is a phase I/II trial of Tinengotinib, that is a new FGFR inhibitor that has been tested in different groups of patients. But what is really important is that Tinengotinib has shown activity in patients previously treated with other FGFR inhibitors and progressing, developing resistance on FGFR inhibitors. And this drug was able to demonstrate clear activity with a very good safety profile in patients previously treated with other FGFR inhibitors. So we can imagine if this data will be confirmed in a large phase III trial that has been planned and is going to start in the next months. If this data will be confirmed, we will be able to define a sequence of different FGFR inhibitors for patients with cholangiocarcinoma and FGFR2 gene fusions.